All right, hello everyone, and welcome back to our next lecture video. Today we're going to focus on the tissues. So the tissues of the human body. Going to look at different tissue types. Sorry, I just realized that I didn't have the sound on. So welcome back to our next lecture. We are going to go over the different tissue types of the human body, and we are going to discuss 4 different types of tissues that we find in biological samples. So let's get started. Histology is going to be the study of tissues, and the study of tissues is going to be what we find the 4 tissue types underneath. Those 4 tissue types are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. A tissue is characterized because it is a similar grouping of cells. So all of these cells in these different tissue types are the same or similar. For epithelial tissue, this is going to be a lining. It's basically going to be lining a cavity or a space that faces something that is exposed to the external environment. Connective tissue is going to be a binding agent, so it will support and also protect other agents and other substances and organs. Muscle tissue is going to be basically functioning for its primary target. If its primary target is the bones, it is skeletal muscle that is moving our bodies physically around. If it is cardiac muscle, it is located within the heart. That is to squeeze and pump the blood out into the circulation. And if it is smooth muscle, it's going to be located in areas of constriction and dilation, such as the blood vessels, intestinal tract, and respiratory tract. Nervous tissue is the type of tissue that we find in electrically excitable cells that send nerve impulses down to target tissues. We have neurons that are the main movers of the electrical current, and we have neuroglial cells, which are supportive cells for neurons. So again, we have our chart here. By the end of our lecture, you should be able to fill in this chart for our 4 tissue types. So to start, we have epithelial tissue. The function of the epithelial tissue is usually going to be 4-fold. It's going to be physical protection, if we're finding it in the skin. It's going to be for selective permeability, allowing things to come across a specific area, such as in the intestinal tract, where we have the lumen contents being transported across the tissue and into the body. Secretions, if we are opening up to a space, we have secretions from glands such as sweat. We have secretions from other glands such as the thyroid or other endocrine glands of nature. And we also have sensation. So we're going to have a neuroepithelial lining. This is going to be in regions for specialized senses such as sight, smell, and hearing. So first things first, we look at the characteristics of an epithelial lining. Epithelium is going to be usually a grouping of cells that is not vascularized. So it's going to be an area with very little or no blood flow. It is always going to be facing a basement surface or basement membrane on one side and a free surface or an apical surface on the other. That free or apical surface could be facing the external environment if it is on the skin, or it's going to be facing the opening of a tube or a lumen or a cavity in the body, such as the opening of the tubules in the kidney. They're going to have intracellular junctions where they're going to be linked between the cells side to side on the lateral surface. They're going to be connected to a connective tissue, some sort of basement substance underneath. And sometimes they're going to have cilia or microvilli on their apical surface, which are going to assist in transportation of molecules across the surface, increase the area for cellular signaling, and also surface area for absorption. Some other characteristics: they're extensively innervated, meaning that they have a lot of free nerve endings that exist. These are for sensations such as temperature, touch, pressure, and pain. As I mentioned previously, they are not vascularized very well, so we have no arteries here. We're going to get nutrients based on absorption. So basically diffusion from the basement membrane is how we get nutrients into these cells. They are usually exposed to a high degree of mechanical or chemical strain. So that strain is going to cause a high regenerative capacity to be necessary. On the surface of the skin, we completely replace the epithelial lining every 27 days. In the intestine, it can be much, much faster, depending on the type of diet, what we expose our intestinal contents to. It can take 3 or 4 days for those to be entirely replaced because of the amount of strain. Epithelium is going to be categorized in either two types of glands. One is exocrine, that's going to be a ductal type of gland, secreting substance to the outside of the body. For example, here we have a sweat gland. So we have chemicals produced by the epithelial lining, secreted into the lumen of the gland, and sent up to the surface of the skin. If it is an endocrine gland, endocrine is going to secrete something into the bloodstream. So that means most of the time we have hormonal secretions. We have a hormonal secretion that is produced by the epithelial lining inside of the endocrine, and we are going to have secretion of those hormones into the bloodstream. And we have some examples of glands over here: pituitary, thyroid, parathyroid, adrenals, kidneys, pineal gland. We can also put the hypothalamus on there. So, types of epithelial glands. Epithelial glandular shape is going to be how they are characterized. So, for one, we have simple tubular. This is a simple lining that is forming a tubular shape or a cylindrical shape. We find these mainly in intestinal cells. We can have a coiled tubular gland. Here, we're going to have a tubular gland that has a coil or a binding at the end. So we're wrapping it around almost like a snail. We find these in American sweat glands. And we have branched tubular. Branched tubular is going to have several layers of branching, as we can see. We find this in the gastric glands, the glands of the esophagus, tongue, and duodenum, which is the initial portion of your small intestine. Simple alveolar. We can say that alveolar glands look like our alveolar sacs, which are the air sacs in the lung. We have a bulb-like ending, so that is an alveolar shape. These are also known as acinar glands. Usually we do not find acinar glands in an adult human. This is usually going to be in a stage of fetal development, and usually they become simple branched glands afterwards. Finally, we have simple branched alveolar glands. So we are going to see that we have several branches, all of saccule or sac-like alveolar shapes. We find this with oil glands that are known as sebaceous glands. And as you see, I have a note down here: an acinar gland is going to be synonymous with alveolar. Compound glands are going to be much more complicated. Compound glands have several different tubes coming off of several different cavities. So we can have a compound tubular. As we see here, we have 3 main branches, and off of those 3 main branches, we have an additional 4 tubes here, an additional 5 tubes here, 4 more tubes here. So there's a ton of surface area for cells to do whatever they're making. We find this in the mucus glands, find it in gastric glands in the stomach, bulbourethral glands next to the prostate, and also the testes. For compound alveolar glands, again, same thing. Here we have 3 branches, and on those 3 branches, we have 3 alveolar shapes. So we have 3 distinct alveolar branches off of each individual tubular branch that has taken its way. So we have a compound acinar gland here. This is seen in a lot of mammary glands. And finally, we have a tubuloalveolar. Right? So this is a combination of tubule shape with an alveolar bulging or bulb at the end. So we find these in salivary areas as well as in respiratory passages and some in the excretory portion of the pancreas, which secretes digestive enzymes. So our types of secretions. Types of secretions, we can have merocrine secretion, which is basically by transport vesicles that release substance out through the top of the cell. We can have apocrine secretions, which is basically part of the cell being pinched off and completely lost into the lumen, or a holocrine secretion. That is where the entire cell itself is actually lysed and apoptosed, and the substances within the cell are released because that cell is bursting and dies. So for merocrine secretion, we find this in salivary glands as well as gastric glands. For apocrine, we find this in mammary glands and the ceruminous glands, which produce sebum, which is earwax. And in the holocrine secretion, we find these in sebaceous glands, which secrete oil onto the top of the skin. Our next is connective tissue. Connective tissue is sort of the stuff that goes in between everything else. So it's going to fill the space where nothing else can fill. It's going to support, protect, and bind all different substances. So our characteristics. They're going to be resident cells that exist inside of connective tissue in most cases. We have fibroblasts, which are going to secrete collagen. We have adipocytes for storage of fat. We have mesenchymal cells, which are going to act in immune functionality. And we also have mixed macrophages. In areas of connective tissue, we are going to have usually a decent amount of vascularity, but it's going to be subjective for what the subtype actually is. A denser, more regular connective tissue is going to have less vascularity than a loose areolar connective tissue. It is not highly innervated by nerves, so we don't really have a regenerative capacity for nerve impulse. And we do not really have a good regenerative capacity for tissue regrowth. So we can think of our example of scar tissue. Scar tissue forms, it is usually tough, hard, collagenous tissue. And this is now connective tissue. So our fibroblasts are the most abundant cell in our connective tissue proper. They're going to produce the fibers of the connective tissue, such as collagen and elastin. We have adipocytes, which are fat cells that will grow or diminish based on the amount of fat that they are storing. We have the mesenchymal cells that are activating as stem cells. They work to replace damaged cells that exist inside of the connective tissue. And we have the fixed macrophages. The fixed macrophages are going to exist as large cellular eaters, or ones that phagocytize pathogens. And they're going to be derived from our normal monocytes, which circulate in the bloodstream. The fibers that we find, as I stated before, we have the fibroblasts that are going to secrete collagen, reticular fibers, or elastic fibers. Collagen fibers are all derived from the type 1 collagen that we find. Type 1 collagen is thick. It does not have much branching. It's going to be a very thick line of fiber. It's going to have a decent tensile strength, and we find this mostly in dense, regular connective tissue, such as in tendons and ligaments. For elastic fibers, these are going to be made from a precursor that is known as eledin. And that precursor eledin is going to yield itself into elastin. It is named because of its elastic qualities. All right. So the elastin is going to be very distensible. It's going to be able to stretch and relax without compromising its original structure. So we find this in areas of a lot of torsion or a lot of mechanical strain, such as skin, vocal cords, and lungs. Reticular fibers are going to be found in supportive tissue networks. So this is going to be where we have a lot of branching, and we have almost like a web-like shape that needs to be around a certain area or surrounding a certain tissue. It's going to contain type 3 collagen. It's going to be a relatively thin fiber structure, and we can find this in places like the spleen and the liver. Next is ground substance. Ground substance is going to be at the basement membrane of places like epithelial linings. It's going to be non-cellular, and it can exist in several different forms. It can be viscous, like in the form of blood. It can be a semi-solid, which is in the form of cartilage, or it can be in a complete solid, such as that of compact bone. When we see an extracellular matrix, or a matrix that is sitting around the collective population of cells, it is usually going to be connective tissue. And if it is connective tissue, that means that it has both protein fibers of either reticular fibers, elastic fibers, or collagen fibers added to the ground substance of the basement membrane. Now, here we have different membranes in the body, where we might find them and what their function is. Our first one is a mucous membrane. Mucous membranes are going to line passages that are opening to the environment. So areas like the nasal cavity, areas like the pharynx and larynx in the throat. They're going to be in areas of the digestive tract as well. Serous membranes, those are going to line bodily cavities. So we see a serous membrane here around the lungs. This is known as the pleural cavity. If it's surrounding the heart, it will be known as the pericardial cavity. Function of a cutaneous membrane, cutaneous being a layer of connective tissue. Here we find a cutaneous membrane known as the dermis. The dermis is the deep layer of the skin. Finally, a synovial membrane. We're going to find this in large multifunctional joints. So we see here that a synovial membrane is going to line a joint space. It's going to have a cavity which contains synovial fluid. We'll find this in shoulder, elbow, knee, large joints like that. Muscle tissue, we have three types. Skeletal, and that's going to be movement of the skeleton. It's going to be highly striated. Cardiac, that's going to exist only in the heart. And smooth, which exists in all the other spaces that need muscular movement and contraction, but it is involuntary. So smooth muscle is going to be involuntary. We do not control it. Same thing for cardiac, involuntary, we do not control it. Skeletal muscles, however, we do control, so we can move those on our own. They're going to have high vascularity. There is a very high oxygenation because of the high energy usage that muscles have. So we are going to have a large amount of blood flow going here. If it is cardiac or skeletal, it will contain striations. Those striations come from alternating proteins known as actin and myosin. Those are contractile. So you have banding upon imagery. You have a thick chain of myosin and a thin chain of actin, and those are going to alternate light and dark bands in the structure. They are pretty well innervated by nerve innervations. We have motor units and motor neurons that innervate all different tissues here. And as mentioned previously, we have involuntary control of the cardiac and smooth muscle, voluntary control of the skeletal muscle. The only one that is very good at recovering from injury is skeletal muscle. So the regenerative capacity of cardiac muscle is almost none, and the regenerative capacity of smooth muscle is minimal, but it can happen. So we see long, thick, densely nucleated striated fibers here for the skeletal muscle. We see small, slender, tapered-ended cells here with no striations for smooth muscle. And we see long, striated, branched, interconnected, striated cardiac muscle cells here. Our fourth type of tissue, the nervous tissue. Nervous tissue exists in everywhere that has nerve innervation. So we have the main type, which is known as a neuron, which is pictured here. And we have supportive or secondary cells known as glial cells. For a neuron, we have a main portion, which is this area, the cell body, which contains a nucleus and mitochondria. We have small branching, which is known as dendrites. These are connecting to the areas around this nerve, communicating with other nerves in the same area, and also the substance outside of the cell. We have an axon, which can either be myelinated or unmyelinated. This conducts the nerve signal down the path to wherever the target is. So these are going to have high vascularity because they use oxidative metabolism exclusively. So we have to have a lot of oxygen going here. So they're highly vascularized. They have very low regenerative capacity. If we have nerve damage or nerve obstruction, usually the damage is permanent if they are destroyed. And we find the location here to be in the central nervous system, which is the brain and spinal cords, and in the peripheral nerves going to every tissue type. So when you look at epithelium, the epithelial types are different based on their different orientations. So as far as a simple epithelium is concerned, if it is simple, that means that it is singular layer. It is one layer of cells. And if it's an epithelium, it's going to be facing some sort of external or apical surface. So here for a simple epithelium, this is what's known as a simple squamous epithelium, which is a simple layer of flat cells that is going to be on top of some sort of basement surface. Primary functionality we have, all right, since it is a flattened out single cell layer, it is pretty easy to diffuse things across this from an outside source to an inside source. So we find that it does very well in filtration, absorption, and secretion of substance. Here come a couple of examples of where we might find this. In the center we have a duodenum. All right, so this is an intestinal villi. We have this on the surface of the capillary. So that allows for easy things like oxygen and carbon dioxide to move across this single cell to exchange carbon dioxide from the blood with oxygen from the atmosphere. A stratified epithelium is going to have multiple cell layers, and it is going to have multiple types of cells sometimes. So we see that this is in areas of high mechanical stress, such as on the surface of the skin. We have many layers of keratinized cells that are stacked on top of one another, facing the external environment. Here, we have a stratified epithelium that is going from the basement membrane up to the mid portion where it is no longer living. So our specialized function here is that the basement layer or basal membrane is a stem cell of sorts. This is a keratinocyte. The keratinocyte stem cell is constantly reproducing more keratinocytes to replace the ones that are dying at the top. So if we're looking through these, we want to identify whether they are simple or stratified. Well, first of all, this looks like a columnar cell layer. So if we look at this, we can see that the nucleuses look like they might be stacked on top of one another, but they are not. So this is what we would call a pseudo-stratified layer, meaning that there are actually not multiple layers of cells here. It's just one layer of columnar-style cells that look like they're layering on top of one another, but they are not. They are simply tapered more so at one end than the other. For B here, we have a layer of simple cuboidal cells. So this is a simple layer, one cell layer of cube-like cells, so box-like cells. For C here, again, it looks like we have a pseudo-stratified. You see a lot of different nuclei that are existing. They look like they are on top of one another. However, it's just different cells forming the same tissue. Finally, in D, we have a stratified epithelium. This is a stratified squamous epithelium, and this looks like it is the epidermis of the skin. As mentioned in our previous picture, we had a pseudostratified epithelium. Pseudostratified epithelium is going to appear as though it has multiple layers, but it does not. So all of the cells that exist here are going to be connected to the basement membrane. Remember that. There is an important mention here. All of our cells are attached to the basement membrane. There is no layering above that. Every cell that exists here has a connection to this basement membrane. So don't be fooled by the stacked nucleuses, right? As long as a part of that cell is touching the basement membrane, it is a part of this initial layer. So we're going to find this in areas such as the trachea, see the vas deferens, which is carrying sperm away from the testes. So we're going to classify the epithelium by its shape. As I said before, if it is flat, this is known as a squamous epithelium. If it is a box shape or a cube-like shape, it is known as cuboidal. If it is a rectangular, it's known as a columnar or cylindrical shape. Places where we find transitional are going to be in the urinary tract. So places like the bladder or the ureters, the transitional epithelium can change. It can crunch up and form more of a column-like shape if the volume of the bladder is less, and it will flatten out and expand as the volume of the bladder becomes more. So if we can characterize all of these in this chart, that'll be a great way to study for this. So here are some examples of simple. Right? We have a simple squamous lining here. You can see this in this capillary. Here we have a simple cuboidal lining. You can find this in most glands, also in the kidneys. Here we have a simple columnar. You can find this in areas of the intestines. We're going to maximize absorption and surface area for absorption. Now, columnar can either be ciliated or not non-ciliated, meaning that we can either have these cellular protrusions at the top, known as cilia, which are going to help move things along, like we have in the respiratory tract. This is known as the mucociliary escalator. And as we have in the fallopian tubes, these are known as fimbriae. For non-ciliated, we're going to have microvilli. Microvilli are just smaller protrusions at the top of the cell. So the cilia are longer. The microvilli are a little bit shorter. They're there to help with extraction of nutrients from the intestinal lumen to push them across into tissues. So our simple epithelium, for a squamous, it's going to be a very easy layer to pass through. It is flattened out and it's only one cell layer, so it allows for easy diffusion. For cuboidal, they allow for easy secretion and absorption. For columnar, they allow for maximizing surface area in areas of nutrient absorption like the intestine. Also, they can act to create substance, such as mucus and digestive enzymes in the pancreas. If we have a stratified, areas of stratified, we have examples of cuboidal within sweat glands. We have a non-keratinized squamous and a keratinized squamous. The non-keratinized squamous is going to be outside of the skin. So we're going to find this in places like the mouth and the vagina. Keratinized squamous is going to be on the top of the skin. So we see the epidermis of the skin here. We have a keratinized layer of squamous epithelium here. Finally, stratified columnar, we do find this. It is very rare because columnar cells are so cylindrical and tall in the first place. Usually we do not find multiple layers stacked upon each other, but you can see them in some apocrine or merocrine sweat glands in some various places. So our function for our stratified epithelium, the squamous, is going to be at the top of the skin to protect against abrasion. Cuboidal, again, it's going to mainly be a protective function. And columnar is going to be either for secretion or protection again. So we see a main theme here that the stratified is multiple cell layers, so it can have a lot of density, and that density allows for it to undergo mechanical strain and stress without compromising too much of its integrity. Transitional epithelium is going to be found in the urinary system. So we find this in the kidneys, sorry, not the kidneys, in the bladder and in the ureters. And this is going to allow for volume expansion. So the function is going to be stretching. If we have an empty bladder, for example, our epithelium here is collapsed. If that bladder is now full, all of a sudden, those cube-like, column-like cells are now flat again. They have become flattened out and they look more squamous in nature to accommodate for the fluid volume, which is filling the bladder. So we see that during a collapsed phase or an empty bladder, we have a more rounded cell surface and they're protruding up into this intracellular space and into the lumen of the bladder. And as the bladder fills, those cells condense, flatten out, and allow for much more surface area to hold fluid volume. So here's our organization and relationship of that. Remember that our simple epithelium comes in squamous, cuboidal, columnar shapes. If it is a pseudostratified columnar, that means it looks like it has multiple layers, but each one of those cells contacts the basement membrane, so it is just one. If it is a stratified epithelium, it has to have 2 or more layers of cells. If it is squamous, it's going to be multiple layered. If it is keratinized, it is at the surface of the skin. If it is non-keratinized, it can be in other surfaces, such as the mouth and the vagina. We have a stratified cuboidal, right? This is 2 or more layers of cube-like cells. Stratified columnar, 2 or more layers of tall cylindrical cells. This is very rare. And transitional. So transitional existing in the bladder and the ureters. These are multiple cell layers of varying shape. Depending on the environment that they're in, the amount of fluid volume that's there, they will either be more squamous-like or more cuboidal-like. All right, so for connective tissue, functionality, physical protection, they're a supportive framework, and they can bind structures together. The physical protection is in areas like the bone, so we have the skull. Supporting and structural framework, the bone yields a place for muscular attachment. For a binding structure, we have ligaments. Ligaments connect bone to bone. We have tendons. Tendons are connecting muscle to bone. If it is dense irregular tissue, this is going to anchor skin to the muscle and bone. We also see connective tissue in storage areas, such as in the adipose tissue and the primary reserve for calcium and phosphorus in the bone. Our blood is a type of connective tissue, and the blood is a connective tissue for the entire body. It's going to carry nutrients, gas, waste, everything else around the body. We have functionality of immune protection. We have leukocytes for the white blood cells that protect against disease. We have extracellular matrix, places that have macrophages and different things like that, lymphocytes, all the other types of immune fighting cells that are going to exist in a connective tissue space. So our types of connective tissue, we have loose connective, which is going to be categorized as either areolar, adipose, or reticular. We have dense connective, which is characterized as either regular, irregular, or elastic. We have supportive, which is mainly cartilages. This is hyaline, fibroelastic, or bone. And we have the fluid, which comes in 2 flavors, blood and lymph. All right, the blood is its own fluid connective tissue. Lymph is derived from the plasma of the blood. All of these are going to be derived from what we call mesenchyme or mesenchymal stem cells. So here is our chart for these connective tissue types. Connective tissue proper is either going to be loose or dense. Loose is going to be what you would think it would be. For being loose, it's going to have fewer cells. It's going to be more sparse. It's going to have more ground substance. It's going to look more fluid. For dense, it's going to have very tightly packed cells. It's going to have more protein, more oomph, more substance, and it's going to have less ground substance there. So loose is going to be more supportive. Dense is going to be stronger, have more tensile connection. So areolar is an example of loose. We have fibroblasts here. Functionality is to bind the skin to deep tissue, surround and protect different organs. It's going to have collagen and elastin fibers there, and it's going to be highly vascularized. For adipose, this is fat tissue. So here we have adipocytes, which are fat cells. They're closely packed together. You see that the nucleuses have been pushed to the periphery of the cell. It's highly vascularized, a lot of blood flow. Main functionality is to store adipose, which is fat cells, for energy, and also to cushion and protect organs. We have reticular. Reticular are going to be very vastly scattered. It's going to be made up of reticular fibers, and it's going to look like a meshwork or almost like a lattice work matrix of fibers put together. Our cell types here are going to be fibroblasts, which secrete collagenous and reticular fibers, and leukocytes, which are going to be the resident immune cells. So we find this a lot in immune functioning organs, such as the thymus, lymph nodes, and bone marrow. Dense regular. Dense regular is going to be in areas like tendons and ligaments. It's going to be very strong, very tensile. It's going to be tightly packed, and it's going to have a cylindrical, organized structure. We're going to have mainly collagen here. Our cell type is fibroblast, which secretes that collagen. It's going to be found in attachments from muscles to bone, as well as from bone to bone. For the irregular connective tissue, this is going to look a lot less organized, a lot more disheveled. We still have collagen fibers and we still have fibroblasts that make them, but it's going to look much less impressive. Still highly vascularized. It's there for support. It's going to be located in the areas of the dermis of the skin and also in the perichondrium of the cartilage. Elastic is going to be the most extensible, so it allows for the most stretch. It is going to be made up of mostly elastic fibers. It's going to be located in the areas of blood vessels that need a lot of distension, like the aorta, also in areas of high torsion and high mechanical tension, like the trachea and the vocal cords, that need to be able to stretch and recoil and reform their initial shape pretty easily. Supporting connective tissues, we have cartilage, which is going to be more flexible, and it's going to be made of collagen and elastic fibers. The cells that make cartilage are known as chondrocytes, and eventually, once they have secreted enough cartilaginous matrix, they will be locked into place in between the cartilage in areas known as the cunei. For the bone, we are similar to cartilage here, except for we have more solidity. So we are more supportive. It is made of collagen, but also made up of our salts. So our inorganic calcium salts of calcium and phosphorus, that gives us this calcification that makes our mineralized solid bone. The cells that this comes from are known as osteocytes, and they're going to be locked into little cavities known as the cunei as well after they have secreted bony matrix. Cartilage types. Hyaline cartilage, this is the most abundant type in the body. We're going to find this in areas such as the nose, the larynx, the ends of long bones at joint surfaces. It's going to have chondrocytes that secrete cartilaginous matrix, and it provides support and protection. Fibrocartilage, this is going to be the most dense of this cartilage. So we're going to find it in areas of a lot of stress or a lot of weight bearing, such as the intervertebral disc of the spine, the meniscus of the knee. We have parallel collagen fibers, so we have a lot of tensile strength. The elastic is going to be the most extensible of the cartilage. We're going to find this in areas that we can pull and retract very easily, such as the lobe of the ear. Also the epiglottis, which is going to cover the trachea while you're swallowing food or water, and the larynx, where our vocal cords are. So bone is what we know as an osseous connective tissue. Compact bone is going to be the outer portion. It's layering the outer layer of the bone. Spongy bone, or what is known as cancellous bone, is going to be on the inner surface, and it is going to be interwoven with red bone marrow. So our bone, the osseous portion, we have osteons here. We can see these little dark circles are osteocytes in between the cavities. We find that this is going to provide a physical framework for our body. So it provides attachment sites for muscles and allows for our body to move in a set manner. It is also a storage location for minerals that we know as calcium and phosphorus. Inside of the spongy bone region and inside of the medulla, or the middle of the long bones, we have production of blood cells, such as red blood cells, white blood cells. This is a term known as erythropoiesis and hematopoiesis. So here's a layering of that. This is an osteon, which is the singular unit of compact bone. And we see that we do have a decent amount of vascularity running through here. Again, we have an osteon. This is our functional unit of compact bone. We have osteoblasts, which are going to secrete bony matrix and build up on the bone, and osteoclasts, which are going to absorb the bony matrix and reabsorb calcium and phosphorus so they can be dispensed back into the bloodstream. All right, spongy bone, also cancellous bone. Mainly we have the interconnection between this and the red bone marrow as our important delineation here for the difference between that and compact bone. All right, and finally we have blood, which is the fluid connective tissue. Again, here we have the formed elements of the blood, which are the red blood cells, white blood cells, and platelets, known as the erythrocytes, leukocytes, and platelets. So it's organized in a solution in terms of the fluid. The red blood cells, white blood cells, and platelets are going to be sitting in the fluid as a suspension. So they're going to be suspended within the plasma of the blood. Main function is transporting everything around the body, whether that be gases, whether that be waste products, whether that be nutrients, hormones, and also functioning in blood clotting if we have any injury to a vascular space. All right. Finally, to recap, if we remember our muscle tissue comes in 3 separate types: skeletal, cardiac, and smooth. Skeletal is going to be voluntary. Cardiac and smooth are involuntary. Skeletal and cardiac have alternating striations, which are the light and dark bands. Smooth muscle does not have striations. So, skeletal muscle here, you see it's very tightly and densely organized. We have dark banding, which is known as your myosin protein, and we have the light banding, which is known as the actin protein. They are long, column-like cylindrical cells with multiple nuclei per cell. They're going to be well organized and stacked on top of each other, almost like the example of lasagna. And they're going to be to move the skeleton. So they're going to be attached to bones of the skeleton. Cardiac, we're going to have similarities to skeletal muscle, except for in this case, we are interconnected. So we are branched between the cells. We have areas known as intercalated discs, or which are also known as desmosomes, connecting the cells together. They're going to be shorter than skeletal muscle, They are going to have striations, but not quite as densely packed, but you can still see some of the striation upon imagery. They're going to be located in the area of the myocardium, which is the middle muscular layer of the heart. And smooth muscle, here we have smaller, slender cells, which usually have tapered ends. They're going to have a single nucleus. They're going to be pretty tightly packed depending on where we are, but they like to be stacked on top of one another. We're going to find this in areas of open tubes. And those open tubes are areas such as the airways, blood vessels, the intestines, and in the uterus, areas where we need to move something through a certain area. We need to propel substance through an organ or through a system. And remember that these are going to be involuntary. All right, and our nervous tissue recap. Final slide here. For our nervous tissue, remember, we have two types. We have neurons and we have glial cells. Neurons have a cell body, central portion with a nucleus. They have dendrites, little spines projecting from that cell body, and they have an axon, which extends for a long distance to its target. Functionality is to send, receive, and process electrical signals and nerve impulses. For glial cells, these are going to be much smaller and much more numerous than neurons. They are there for the protection and support of neurons. For both of these cells, neurons and neuroglial cells, we will find them all over the place where there is neural innervation. The central nervous system and the brain and the spinal cord, and the peripheral nervous system all the way to the edges of the skin with those nerve endings. Alrighty, those are our tissue types. Thank you for listening to our next lecture, and I will see you the next time. Take care.